Welcome to part 1 of lecture 13 on the endocrine system. Today we're going to talk about an overview of the endocrine system. We'll also discuss the types of hormones. So this chapter is back a little ways in your textbook. This is chapter 16. Let's begin with an overview of the endocrine system. And I really like the definition that's on the right side here because this gives you a lot of information about the endocrine system. The endocrine system, as it says, is a collection of cells and tissues scattered throughout the body that produces substances released into the blood. These are the hormones. Ultimately, these affect the activity and the metabolism of target cells. Not every cell in the body, but target cells. And we're going to talk about exactly what makes a cell a target cell a little later. One of the things you'll notice is that we have many tissues scattered throughout the body, some of which we've already talked about, maybe not in an endocrine capacity, but we've talked about, for example, the heart. We've talked about the digestive tract. We've talked a little bit about the thymus when we talked about the immune system. So some of these tissues we've talked about already, but we're going to cover their endocrine functions during this series of lectures. Now we're going to eventually get to all these tissues and talk about what they make, how the hormones act, and very importantly, how hormone release is controlled. The next thing I want to do is talk a little bit about the differences between the endocrine system and the exocrine system. Endocrine glands are ductless. That means they do not have ducts. If we compare exocrine glands, you notice that these do have ducts, and you can see an example of a duct of an exocrine system on the right. So this passageway into which the cells put their secretions is the duct. This will eventually go onto a free surface and exert its effect extracellularly. That is, the effects of exocrine gland secretions are exerted outside cells. However, if you take a look on the upper left again, you'll see that the effects of hormones are intracellular. As we said before, hormones affect the internal activity of cells. They regulate their metabolism, activity, and we'll talk about those things in a couple of minutes. Now, if we look at one of the endocrine glands like the thyroid gland and take a close-up, you'll notice that these cells that are in the thyroid gland are secreting their hormones directly into the blood. Now, I just want to remind you about something that we said in A&P1. When we say directly into the blood, we're not talking about the fact that cells are directly connected with the blood vessels at all. So over here would be a blood vessel. Over here, of course, would be a cell. And this, remember, is the interstitial fluid. Now, when we say that these cells secrete hormones directly into the blood, basically what happens is they secrete the hormone into the interstitial fluid, and then that hormone goes into the bloodstream, into a blood vessel. So it really is a two-step process. But the reason we say that they secrete the hormones directly into the blood is because they do not go through a duct before they get into the bloodstream. Now, as we said, endocrine glands release hormones that travel throughout the body to target cells. And very importantly, to make something a target cell, it must have a receptor in order to respond. So we're talking about, for example, a cell like this that might have a receptor somewhat shaped like this. Another one that has a receptor shaped like this, another one that might have a receptor shaped like this. And if we have a hormone molecule floating by here in the interstitial fluid, which one of these is it going to bind to? Sure, it's going to bind to the one that's complementary shaped to its own shape. And so these hormones can only bind to receptors that are designed to fit them specifically. And as we'll see, once these hormones bind, they then cause some kind of activity to happen inside the cell. As we said, hormones act intracellularly. Now, in addition to endocrine glands, which release hormones that travel throughout the body in blood, we also have a couple of other types of secretions in the body. One is known as paracrine secretion. This is a local type of secretion. Things, for example, like histamine, nitric oxide, somatostatin that acts in the pancreas to downregulate secretion of insulin and glucagon, eicosanoids, which we're going to talk about in a couple of slides. All of these things act locally. In other words, they do not travel through the blood. We also have secretions that we can term autocrine. This means that one cell secretes something that affects the same cell that secreted it. This is a major mechanism that lymphocytes of the immune system use to control their activity, and this is called autocrine secretion. Now, as I said before, hormones regulate metabolic processes and change activities of cells. Some of the things that we're talking about include these. For example, rates of biochemical reactions within cells. These can either speed up or slow down reactions. They regulate water and electrolyte balance. very important in the body. We're going to come to those lectures a little bit down the road. They also can regulate blood pressure, as we'll see, and they're very important in reproduction, development, and growth. All of these are intracellular processes. In terms of a comparison of the nervous system and endocrine system, you'll see in these two figures below that when nerves secrete a neurotransmitter, the effect is more of a paracrine type secretion. In other words, these neurotransmitters only have to go a very short distance in order to affect the cells that are their targets. Very little of this gets out into the blood. Occasionally, a neurotransmitter does seep into the blood, get absorbed, and maybe travel throughout the body, but its concentrations are generally so low in the blood that it really doesn't have a systemic effect. On the other hand, when we look at endocrine glands, their primary purpose is to secrete hormone into the blood. And once the hormone is in the blood, it gets everywhere in the body. Now, as far as the cells that the hormone acts on, remember that target cells must have specific receptors to bind the hormone. So as you see here, not all hormones will act on all the cells in the body. Only the cells that have receptors for it will be affected and have their intracellular activity affected by that hormone. So let's talk a little bit about the chemistry of hormones. And to do that, we want to first classify the hormones and break them down into a couple of major groups. So we can take the hormones and we can divide them chemically into amino acid derivatives. These are going to include hormones that are like amino acids, small groups of amino acids together, these are peptides. Proteins and glycoproteins are also included in this group. All of these are amino acid derivatives. And then we have another group that are cholesterol derived. These are the steroids, like the sex hormones, for example, estrogen, testosterone, like some of the adrenal gland hormones. So we're going to talk about those a little bit later. And finally, we have a group called eicosanoids. Now, these are paracrine type factors. They act locally, but they're very important. So I'm going to discuss them just a little bit, and then we're going to leave them for the rest of the lectures. We're really only going to be concerned in the endocrine system with amino acid derivative hormones and with steroid hormones. But let me just talk a little bit more about the eicosanoids since they are so important. As you see here, eicosanoids are also lipid derived along with the steroids. Eicosanoids are important paracrine factors that mediate many processes in the body, as you see here. And this list is by no means inclusive, but some of the processes include inflammation, blood vessel constriction, blood clotting, smooth muscle contraction and relaxation. And the way eicosanoids are produced is by using the lipids inside the cell membrane. These phospholipids from the cell membrane are converted into something that's known as arachidonic acid, which is a starting point for the eicosanoids. And this conversion is catalyzed by an enzyme known as phospholipase A2 that's in tissues. These are called eicosanoids, eicosa meaning 20. These are typically 20 carbon-containing compounds. Now, depending on the enzyme that arachidonic acid is exposed to, the eicosanoids can be produced by two different enzymes. One is called lipoxygenase, the other is cyclooxygenase. As you see, we can make things with lipoxygenase like leukotrienes. These are very important immune cell and leukocyte type mediators. We can make things with cyclooxygenase like thromboxanes, prostacyclin, prostaglandins. We've talked about thromboxanes and we've talked about prostacyclins before. Remember, thromboxanes are things that cause smooth muscle contraction in the blood vessels and cause blood vessel constriction. Prostacyclin are those compounds secreted by endothelial cells that make platelets glide a little bit more easily across the endothelial lining. Prostaglandins in general are a class of molecules that do many different things in the body. One of the things a certain class of prostaglandins do is to cause pain. So basically, these substances are very important because what they do is they create conditions that many times must be managed clinically. For example, pain, chronic inflammation. So later, when many of you study pharmacology, you'll come back to the eicosanoids because this is a big pivotal point in patient management, pain management, and disease management. This table is a good summary of the different types of hormones that we have. You'll notice this dashed line here that separates the two major classes of hormones. These are amino acid derived hormones, and the ones on the bottom, of course, are derived from cholesterol. Now, if I ask you examples of these hormones, I think a better strategy for remembering examples of these is to remember the steroid hormones, since there are far fewer of those. And then if a hormone that I give you is not a steroid, it's going to be an amino acid derived hormone. So some of the steroid hormones include, as I said, estrogens, testosterone, aldosterone, cortisol, These are hormones that are secreted by the adrenal gland. We'll talk about those. These are hormones on the top that are secreted by the gonads, that is by the testes or by the ovaries. So let's talk a little bit about the chemistry of these different kind of hormones. First, we'll talk about steroid hormones. Steroid hormones, as we said, are derived from cholesterol. This four-ring structure, as you remember from AMP1, is a cholesterol nucleus. And depending on the things that we stick on here, which are called substituents, it will change the chemistry of the molecule. This particular molecule that we're looking at is cortisol. Now, steroid hormones are hydrophobic, as you would guess, since they're synthesized from cholesterol, which is a fat. So this makes them lipid soluble. So anytime we talk about the chemistry of hormones, we also have to talk about the meaning for blood plasma solubility and cell membrane solubility. A hormone like cortisol would not be very soluble in the plasma because this is a hydrophobic hormone, and the plasma, as we said, is mostly water. On the other hand, since cell membranes are largely fatty material, cortisol would dissolve, so to speak, in the cell membrane very easily, and we would find that cortisol crosses the cell membrane very, very easily. Let's take a look at the action of steroid hormones. We're still focusing on steroid hormones, and let's take a look at how they act. Now, this hormone molecule that you see here is a steroid hormone, and you'll notice the cholesterol nucleus. So we'll take this just as a general class of steroid hormone. You'll notice that it goes right through the cell membrane and does not have to bind anything or have any help crossing the cell membrane. So the first step is that the hormone molecule will cross the cell membrane. Next, what happens is the hormone combines with a receptor in the nucleus or cytoplasm. You'll remember from what we said earlier that in order for a cell to be affected by a hormone, to be a target cell, it must have a receptor for that hormone. So cells that have hormones come into the cell are no different. They also need receptors for these hormones in order to have some kind of activity change. So the hormone comes across the cell membrane, comes into the cell. Sometimes there are receptors in the cytoplasm. In this particular case, we're looking at intranuclear receptors that are inside the nucleus. You'll notice the hormone molecule here and the receptor molecule here. What we form when the receptor binds to the hormone is a receptor-hormone complex, or a hormone-receptor complex. Now, very importantly, what happens is this hormone-receptor complex, not each individual part of it, will cause an increase in some kind of messenger RNA synthesis. This is, of course, going to lead to some type of protein synthesis. So the messenger RNA goes to the cytoplasm. This will eventually produce a new protein molecule. And we can use the example of something called aldosterone that's secreted by the adrenal gland. This is a protein that will eventually get put back in the cell membrane here, and its purpose is as a sodium-potassium pump. In other words, this is going to regulate the water concentration of cells. And this is the mechanism of aldosterone action. It comes through the cell membrane, binds to an intranuclear receptor. The hormone receptor complex then binds to DNA, which produces mRNA. The mRNA gets translated into new protein, and that protein gets placed in the membrane and then can regulate the water balance of the cell. Now, oddly, thyroid hormone is actually an amino acid-derived hormone. However, it has a similar mechanism of action as steroid hormones, even though it is derived from an amino acid, specifically tyrosine. So we do need some proteins to bind to thyroid hormone and carry it in the plasma, since it's not really very soluble. So what we can say about steroid hormones is that the magnitude of cellular response, the change in cellular activity, is proportional to the number of hormone receptor complexes formed. I think you can understand this. If we have, for example, 1,000 hormone molecules, but I only have one receptor in the nucleus, I can only form one receptor hormone complex at a time. And so the level of activation inside the cell is going to be very low. By contrast, if I had one hormone molecule, but 1,000 receptor complexes inside the nucleus, I could still only form one hormone receptor complex. Level of activity would be very low. What ultimately happens in the cell really depends on the number of hormone receptor complexes formed, and the response from the hormone is proportional to the number of those hormone receptor complexes. Let's turn our attention now to amino acid-derived hormones. Here you see some examples of these, like norepinephrine, parathyroid hormone, oxytocin, and you see that these can vary greatly in size. The main commonality is that they're all derived in some way, shape, or form from amino acids, which is why we call them amino acid-derived hormones. Now, some of the characteristics include that these are in general water-soluble or hydrophilic, so they get along very well in the plasma of the body. However, since cell membranes are largely hydrophobic, they do not have an easy time of crossing the cell membrane. They have to find a different way to activate the cells than what we saw for steroid hormones. The very first step, as you see out here, is that the hormone molecule has to, as always, bind to a receptor. Notice where the receptor is for this class of hormone. These receptors are in the cell membrane. And what the cell membrane actually is going to do is going to pass along the message of the hormone to the inside of the cell. So let's take a look at how this works. The first thing is we call the hormone a first messenger, as you see here. This is going to deliver the message of the endocrine gland to this particular target cell. And so the hormone has to bind in a receptor inside the cell membrane. As you see, this is a pretty good fit. So we have a receptor that's complementary in shape to the hormone. Once this hormone is bound, what this does is it activates an enzyme inside the cell. In this case, it's adenylate cyclase. And the purpose of this enzyme is actually to convert ATP into something called cyclic AMP. Now, cyclic AMP is a very important messenger molecule inside cells, and so we call cyclic AMP a second messenger. So in other words, this is going to take the original message by the hormone and translate it into some activity inside the cell, and for that reason we call it a second messenger. If you look at the statement on the bottom of the slide, you'll see that the magnitude of response for amino acid-derived hormones is not directly proportional to the number of hormone receptor complexes, it's amplified. So let's see how this works. We have one hormone molecule in this example binding to one receptor. Let's imagine that we have 100 molecules of ATP that are converted into 100 molecules of cyclic AMP. And in turn, let's say that each cyclic AMP molecule could cause 100 conversions that we see in the step 5 here. In other words, a protein kinase that's inactive is now being activated by each of these cyclic AMP molecules. And let's imagine that each of the protein kinases that becomes activated can now activate 100 substrates of some sort. That would be an effect inside the cell of 100 times 100 times 100. Each of these, of course, is 10 to the second power. So 10 to the second times 10 to the second times 10 to the second is 10 to the sixth, or 1 million. So what we say is that this one hormone molecule activated 1 million of these substrate molecules. So I think you can clearly see that this is an amplification. Just due to one hormone receptor complex forming, we have 1 million of these substrate molecules active. Now inside the cell, of course, these numbers are usually much larger. So we have a great amplification here. And this is a little bit different mechanism than we saw for the steroid-derived hormones. Now let's talk a little bit more about target cell activation by hormones. We said before that target cells must have specific receptors to bind the hormones. And the target cell activation itself depends upon several different things. One is the blood levels of the hormone. In other words, how much of this hormone do we have in the blood? The second is the relative number of receptors for this hormone. And then finally, we have something called affinity, or strength of binding of the hormone. Let's talk a little bit about blood levels of the hormone. The hormones are released by an endocrine organ, and the rate of release from an endocrine organ can vary. There are some circumstances in the body where the rate of hormone release is elevated. There are some circumstances where the rate of release is decreased. So the rate of release will determine the blood levels of the hormone. We also have hormones that are degraded by different cells in the kidney, for example, in the liver. These are very good at breaking down the hormones to inactivate them. So as you can imagine, much of this depends upon the release of the hormone, how much of it is put into the blood, and then at the same time, how much of this is degraded. The difference between these two will basically determine what's called the hormone's half-life. So remember, we usually abbreviate this as T1 half. We're basically talking about the time for half of the activity of a molecule to dissipate or disappear. So we use half-lives in terms of maybe hours, minutes, days sometimes. And we say that at the end of each half-life, half of the activity of that particular molecule has dissipated. Now the second factor are a relative number of receptors for the hormone. We said before that especially for steroid-type hormones, there's a direct proportionality between the number of receptor hormone complexes that we form. The same can also be said of amino acid-derived hormones. The more receptors that we have in the cell membrane, the more of those molecules inside the cell we can activate. And the number of cell receptors for the hormones can actually be up or down regulated depending on the needs of the body. And finally, we have something that's known as affinity or strength of binding of the hormone to its receptor. If I had a receptor on a cell that was shaped like this and I had a hormone molecule shaped like this hormone could pretty much fit very nicely in here. If the receptor was shaped a little bit differently, maybe like this hormone could still fit in here, but the binding wouldn't be very tight. It'd be likely that this would bind and then come away as fast as it bound. So the affinity for this is much higher than for this. So anytime a hormone binds with high affinity to its receptor, it will generally stay in the receptor for a longer time and activate more of whatever that hormone does inside the cell. So that finishes off part 1, our introduction to hormones, a little bit about the chemistry of hormones. I'll see you very soon for part 2 of the endocrine system, and we'll start talking about some of the tissues and some specific hormones. See you then.